Today, I will be discussing groundbreaking research from the University of California, San Diego that sheds light on various aspects of human health, from tackling complications related to diabetes and cancer to addressing debilitating conditions like facial paralysis. These studies, conducted by leading scientists, have unveiled new insights into the role of insulin in our bodies, the mechanisms behind viral infections leading to cancer, and innovative treatments for a range of medical conditions.
Researchers at the University of California, San Diego have found that ultraviolet UV nail polish drying devices used for curing gel manicures can damage human cells, leading to cell death and cancer-causing mutations. The study revealed that a single 20-minute session using these devices led to 20–30% cell death, while 3 consecutive 20-minute exposures caused 65–70% cell death. UV exposure also resulted in mitochondrial and DNA damage, leading to mutations associated with skin cancer in humans. Although long-term epidemiological studies are needed to confirm the risk of skin cancer from these devices, the study highlights potential health concerns related to their chronic use. Epilepsy affects a significant portion of the population, with 4% of people experiencing it, especially children. While modern medicine can control most seizures, approximately 20% of patients do not respond to treatment, and some may have malformations of cortical development, MCD, in their brains. Surgical removal of these abnormal brain tissue patches can cure seizures, but the causes of these patches have been elusive. Researchers from the University of California, San Diego School of Medicine and Rady Children's Institute for Genomic Medicine, along with an international consortium of children's hospitals, have made a significant breakthrough in understanding the genetic causes of MCD. They identified 69 different genes with somatic brain mutations, most of which have not been previously reported in MCD. These findings could lead to new classifications and personalized therapies for epilepsy related to MCD. Researchers from the Salk Institute and the University of California, San Diego have discovered a link between altered amino acid metabolism and peripheral neuropathy in diabetic mice. Peripheral neuropathy, which affects approximately half of individuals with diabetes, causes weakness, numbness, and pain in the hands and feet due to damage to peripheral nerves caused by high blood sugar levels. The study found that low levels of the amino acid serine can lead to the accumulation of atypical sphingolipids, contributing to nerve damage. Serine supplementation in diabetic mice slowed neuropathy progression, and a compound called myriocin, which inhibits the enzyme responsible for the amino acid switch in sphingolipids, reduced neuropathy symptoms. While serine-rich foods and supplements are available, the researchers caution against using them until more research is conducted to understand the potential benefits and downsides of supplementation. Researchers led by Dr. Don Cleveland at the University of California, San Diego have made significant progress in understanding and potentially treating amyotrophic lateral sclerosis, ALS, Alzheimer's disease, AD, and frontotemporal dementia. In these diseases, the protein TDP-43 is lost from the nucleus of cells, leading to the loss of another protein called stathmin-2, which is crucial for neuron regeneration and maintaining connections to muscle fibers. The study shows that designer DNA drugs can restore normal processing of RNA encoding stathmin-2, preventing its loss. Mouse models engineered to mimic this condition were treated with designer DNA drugs, leading to the restoration of stathmin-2 levels. Clinical trials may be on the horizon to delay paralysis in ALS patients by maintaining stathmin-2 protein levels using these designer DNA drugs. Steve Chafe, a firefighter diagnosed with a rare benign skull-based tumor that affected his facial nerves, experienced facial drooping and paralysis on the right side of his face following tumor removal. A specialized team at UC San Diego Health, including facial nerve surgeons and physical therapists, performed a series of delicate surgeries and therapies to restore his smile and facial function. The procedures involved nerve transfers, grafts, and a gracilis-free muscle transfer, connecting tiny nerves and vessels. Chafe's successful recovery has brought back his spontaneous smile, and he now lives a fulfilling life with his family and a service dog. UC San Diego Health's Facial Nerve Clinic specializes in comprehensive care and research for individuals with facial paralysis. The Epstein-Barr Virus, EBV, a common virus infecting over 90% of the global population, has been linked to various diseases, including cancer. Researchers from the University of California, San Diego have discovered how EBV exploits genomic weaknesses to cause cancer and reduce the body's ability to suppress it. The virus induces cleavage of human chromosome 11, initiating genomic instability that can activate leukemia-causing oncogenes and inactivate tumor suppressors. This study sheds light on how EBV contributes to cancer development and provides insights into potential prevention and treatment strategies by targeting EBV-induced chromosomal abnormalities. Researchers at the University of California, San Diego have discovered a key player in preventing insulin shock, a potentially deadly condition caused by excessive insulin levels. They identified a metabolic enzyme called fructose-1, 6-bisphosphate phosphatase, FBP1, as the body's natural defense against insulin shock. FBP1 not only regulates gluconeogenesis, a process that synthesizes glucose in the liver, but also inhibits the protein kinase AKT, a primary conduit of insulin activity. The researchers developed a peptide called E7 derived from FBP1, which has the potential to be used as an alternative to insulin, as it doesn't induce insulin shock. This discovery could lead to safer and more effective treatments for diabetes.